Today we are going to learn about some aspects related to innate and adaptive immunity, B cell and T cell. So first, let's learn what is innate immunity. Innate or non-specific immunity is the defense system with which you were born. It protects you against all antigens. Innate immunity involves barriers that keep harmful materials from entering your body. These barriers form the first line of defense in the immune response. Examples of innate immunity include cough reflex, enzymes in tears and skin oils, mucus which traps bacteria and small particles, skin, stomach acid. Then innate immunity also comes in a protein chemical form called innate humoral immunity. Examples include the body's complement system and substances called interferon and interleukin-1, which causes fever. So pathogen enters, sensors are there, signal is there, pattern recognition will be taking place, internalization, then cytokines or interferons are released. And cells and molecules of innate immunity. Physical and chemical barriers such as skin and antimicrobial chemicals produced at epithelial surface. So physical and chemical barriers such as skin and antimicrobial chemicals produced at epithelial surface. Soluble effector proteins including components of complement system and other mediators of inflammation. Cellular components such as phagocytic cells like neutrophils, macrophages, NK cells and other innate lymphoid cells. Then cell-associated pattern recognition receptors and sensors. And group of serum protein increase in concentration in response to infection are termed as acute phase proteins. C-reactive protein, serum amyloid protein, mannose binding protein, etc. CRP, MBP, SAA. These proteins maximize activation of the complement system and opsonization of invading pathogens. Physical and chemical barriers provided by skin and mucous membrane. Chemical barriers at surface include specialized antimicrobial proteins and peptides. At epithelial surface, lysozyme, lactoferrin, defensin. Lysozyme tears cleaves the peptidoglycan components of bacterial cell wall. Lactoferrin binds and sequesters metal ions needed by bacteria and fungi, limiting their growth. Then defensin are cysteine-rich cationic peptides help in killing phagocytosed microbes. Then cellular components. There are many types of white blood cells or interleukocytes that work to defend and protect the human body. In order to patrol the entire body, leukocytes travel by way of the circulatory system. Phagocytes or phagocytic cells. Phagocyte means eating cell, which describe that role phagocytes play in the immune response. Phagocytes circulate throughout the body looking for potential threats like bacteria and viruses to engulf and destroy. You can think of phagocytes as security guards on patrol. Then macrophages. Macrophages, commonly abbreviated as macrophages, are efficient phagocytic cells that can leave the circulatory system by moving across the walls of capillary vessels. The ability to roam outside of the circulatory system is important because it allows macrophages to hunt pathogens with less limits. Macrophages can also release cytokines in order to signal and recruit other cells to an area with pathogens. Then mast cells. Mast cells are found in mucous membranes and connective tissue and are important for wound healing and defenses against pathogens via the inflammatory response. When mast cells are activated, they release cytokines and granules that contain chemical molecules to create an inflammatory cascade. And mediators such as histamine cause blood vessels to dilate, increase blood flow and cell trafficking to the area of infection. The cytokines released during this process act as a messenger service, alerting other immune cells like neutrophils and macrophages. Then neutrophils. Neutrophils are phagocytic cells that are also classified as granulocytes because they contain granules in their cytoplasm. These granules are very toxic to bacteria and fungi and cause them to stop proliferating or die on contact. Then eosinophils. Eosinophils are granulocytes target multicellular parasites. Eosinophils secrete a range of highly toxic proteins and free radicals that kill bacteria and parasites. The use of toxic proteins and free radicals also cause tissue damage during allergic reactions. So activation and toxin release by eosinophils is highly regulated to prevent any unnecessary tissue damage. Then basophils. Basophils are also granulocytes that attack multicellular parasites. So basophils release histamine much like mast cells. The use of histamine makes basophils and mast cells keep players in mounting an allergic response. Then natural killer cells. Natural killer cells, NK cells, do not attack pathogens directly. Instead, natural killer cells destroy infected host cells in order to stop and spread of an infection. Infected or compromised host cells can signal natural killer cells for destruction through the expression of specific receptors and antigen presentation. Then dendritic cells. Dendritic cells are antigen presenting cells that are located in tissues and can contact external environments through the skin, the inner mucosal lining of the nose, lungs, stomach, and intestine. Since dendritic cells are located in tissues that are close common points for initial infection, they can identify threats and act as messengers for the rest of the immune system by antigen presentation. Then cell-associated pattern recognition receptors and sensors. Cells and many soluble proteins have the ability to recognize pathogen-associated molecular pattern (PAMP) for initial detection of pathogens. Different types of pathogen express different PAMPs. Innate immune system also recognize endogenous molecules that are produced by or released from damaged or dying cells called the damage-associated molecular patterns (DAMPs). Innate immune system have different types of receptors in different locations to recognize PAMPs and DAMPs. PAMPs are recognized by pattern recognition receptors, PRRs. Macrophages, neutrophils, dendritic cells express greatest number of these receptors. PRRs linked to intracellular signal transduction pathways that activate various cellular responses. Mammals have several distinct classes of cell-associated PRRs using tool-like receptors, RIG1-like receptors, node-like receptors, AIM2-like receptors, and C-type lectin receptors. TLRs were first identified and are best characterized. So toll-like receptors are a class of pattern recognition receptors that function exclusively as signaling receptors. Identified as a protein in Drosophila melanogaster embryo for developing dorsoventral polarity. In adult fly, it acts as fungal infection resistance. TLR1, integral transmembrane glycoproteins, is composed of an extracellular N-terminal domain and an intracellular C-terminal domain. In human, it is 700 to 1100 amino acid long. N-terminal domain is constituted by about 16 to 28 leucine-rich repeats and has the function to recognize PAMP, whereas the second, called toll IL-1 receptor TAR domain, that initiates downstream signaling. So members of TLR family is 10 in human, TLR1, TLR10 and 11 in mice. TLRs localized to the plasma membrane, to the membrane of intracellular compartments such as FR, endosome, lysosome or endolysosome. They recognize distinct or overlapping PAMPs such as lipid, lipoprotein, protein and nucleic acid. TLRs expressed in innate immune system or immune cells such as dendritic cells and macrophages, as well as non-immune cells such as fibroblast cells and EP cells. Based on location, TLR is classified into the subfamily cell surface TLRs and intracellular TLRs. In human cell surface, TLRs are TLR1, TLR2, TLR4, TLR5, TLR6, TLR10. Intracellular TLRs include TLR3, 7, 8 and 9. TLRs and their ligands. TLR2, TLR1, bacterial triacyl lipopeptides. TLR2, fungal phospholipomannan, bacterial peptidoglycans. TLR3, viral dsRNA. TLR4, bacterial lipopolysaccharide, fungal mannan, bacterial flagellar 5. Then toll-like receptor signaling pathways. Toll-like receptors recognize distinct pathogen-associated molecular patterns and play a critical role in innate immune responses. They participate in the first line of defense against invading pathogens and play a significant role in inflammation, So toll-like receptors recognize distinct pathogen-associated molecular patterns and play a critical role in innate immune response. So they participate in the first line of defense against invading pathogens and play significant role in inflammation, immune cell regulation, survival, and proliferation. To date, 11 members of the toll-like receptor family have been identified, of which TLR1, TLR2, TLR4, TLR5, TLR6, and TLR11 are located on the cell surface, and TLR3, TLR7, TLR8, and TLR9 are localized to the endosomal lysosomal compartment. The activation of the TLR signaling pathway originates from the cytoplasmic toll IL-1 receptor TIR domain that associates with the TIR domain containing adapter MYD88. Upon stimulation with ligands, MYD88 recruits IL-1 receptor associated kinase 4 IRAK4 to TRL through interaction of the death domains of both molecules. IRAK1 is activated by phosphorylation and associates with TRAF6, thereby activating the IKK complex and leading to activation of MAP kinase, JNK, P38 MAP kinase, and NFKB. TOLIP and IRAKM interact with IRAK1 and negatively regulate the TLR-mediated signaling pathways. Additional modes of regulation for these pathways include TRIF-dependent induction of TRAF6 signaling by RIP1 and negative regulation of TARAP-mediated downstream signaling by ST2L, TRIA-D3A, and SOCS1. Activation of MYD88 independent pathways occurs via TRIF and TRAF3, leading to recruitment of IKK, kappa, TBK1, phosphorylation of IRF3, and expression of interferon beta. TIR domain containing adapters such as TIRAP, TRIF, and TRAM regulate TLR mediated signaling pathways by providing specificity for individual TLR signaling cascades. So TRAF3 plays a critical role in the regulation of both the MYD88 dependent and TRIF dependent signaling via TRAF3 degradation, which activates MYD88 dependent signaling and suppresses TRIF dependent signaling. So TLR, recruitment of adapter proteins, recruitment and activation of protein kinase, later activation of transcription factors, gene transcription, TLR and basic signaling mechanisms. Then inflammatory response. Sensor cells serve to amplify immune response by the production of inflammatory mediators. Thus are the secretory proteins called cytokines. Cytokines released by activated sensor cell act to recruit leukocytes and plasma proteins from the blood into infected tissue, a process known as inflammation that helps to destroy the pathogen. So inflammatory response delivers the cells and molecules of host defenses to the sites where infecting agents need to be compacted. Then cytokines are small chemoattractant proteins that stimulate leukocyte movements and regulate the migration of leukocyte from the blood to tissue. Cells of inflammatory response are macrophages and neutrophils. These are inflammatory cells. Leukocyte is recruited first from the blood into sites of inflammation as the neutrophil because it is the most abundant leukocyte in the blood and respond most rapidly to the chemotactic signals. The eosinophils also migrate into inflamed tissue and contribute to the destruction of invading microorganisms. Then cytokines, low molecular weight, less than 30 kilodalton, soluble proteins, non-immunoglobulin in nature, secreted by immune cells. Cells of immune cell system communicate through the exchange of secreted cytokines. Cytokines and hormones secreted in response to discrete stimuli. Cytokines produce action only when they bind to specific high affinity receptors on the surface of target cells. Cytokine receptors are transmembrane protein where the binding of cytokines occur in the extracellular region and intracellular interaction with signaling proteins. There are over 100 different cytokines. They have different names depending either on their origin or their activity. Lymphokines, monokines, chemokines, interleukin, interferons, TNF family. Then amlodipines modulating cytokines are there related to inflammation. Interleukins. ILs are a group of cytokines secreted by a variety of leukocytes. Majority of interleukins are synthesized by Th cells as well as through monocytes, macrophages, and endothelial cells. They promote the development and differentiation of T cells, B cells, and hematopoietic cells. Then chemokines. These are defined by conserved cysteine residue. The name chemokine is due to its ability to induce directed chemotaxis in nearby responsive cells. These proteins activate GPCRs and induce cells to migrate. The role of chemokine is to promote the accumulation of these cells at the source of chemokine production. Then interferon, INFs are involved in antiviral defense, cell growth regulation, and immune activation. It was the first discovered cytokine. Human interferons classified into three major types: type 1 IFN, type 2 IFN, type 3 IFN, and TNF family. They activate the signaling pathways for cell survival, death, and differentiation. It constitutes 19 members that mediate various biological functions. Then hematopoietins. Hematopoietins, cytokines. These factors include the various colony stimulating factor, CSF, including GCSF, GM-CSF, M-CSF, hypoerythropoietin, SCSF, stem cell factor, SCPF, stem cell proliferation factor, and various interleukins. Cytokine, target cell, chemotaxis, apoptosis, metabolic activation, proliferation, differentiation, inhibition of proliferation. Primary cell sources: macrophages, endothelial cells, activated T cells and Th cells, macrophage Th cells, like that. Then adaptive immunity. Adaptive immunity is referred to as acquired immunity or specific immunity, as it is only found in vertebrates. The adaptive immune response is specific to the pathogen present. The adaptive immune response is meant to attack non-self pathogens but can sometimes make errors and attack itself. It may be natural, acquired immunity naturally, and it can be active or passive, or artificial, acquired immunity through medical procedure. Specificity, diversity, memory, specialization, self-limitation, non-reactivity to cell. What is specificity? Ensure that distinct antigens elicit specific responses. Diversity enable immune system to respond to a large variety of antigens. Then memory lead to enhanced response to repeated exposure to the same antigen. Specialization generate responses that are optimal for defense against different types of microbes. Self-limitation allows immune system to respond to newly encountered antigens. The non-reactivity to cells prevents injury to the host during responses to foreign antigens. Differences between active and passive immunity. Active immunity, exposure to antigen. Immunity achieved by injecting antigen. Activation of immune system. Immune states develop over a period of weeks. Immunological memory develops in case of active immunity. But in passive immunity, no exposure to antigen. Immunity achieved by injecting antibody or immune cells. No immune system activation. Immunity develops immediately. No immunological memory develops. Immunity, innate immunity, adaptive immunity. Adaptive in the sense natural immunity, artificial immunity is two types. So this natural immunity is again classified into two, like active immunity, a person developing his or her own immune response after exposure to pathogens. Next, another type of natural immunity is passive immunity. One person receives performed immunity made by another person. Then artificial immunity, active immunity, and passive immunity. Active immunity in the sense a person develops his or her own immune responses after vaccination. Passive immunity, one person receiving performed immunity made by another person. Then cells of adaptive immune system. The adaptive immune system relies on fewer types of cells to carry out its tasks. B cells and T cells. Both B cells and T cells are lymphocytes that are derived from specific type of stem cells called multipotent hematopoietic stem cells in the bone marrow. After they are made in the bone marrow, they need to mature and become activated. Each type of cell follows different paths to their final mature forms. There are two main mechanisms of immunity within the adaptive immune system: humoral and cellular. Humoral immunity, also called antibody-mediated immunity. With assistance from helper T cells, B cells will differentiate into plasma B cells that can produce antibodies against a specific antigen. The humoral immune system deals with the antigens from pathogens that are freely freely circulating or outside the infected cells. Antibodies produced by the B cells will bind to antigens, neutralizing them or causing lysis, dissolution or destruction of cells by lysine or phagocytosis. Cellular immunity occurs inside infected cells and is mediated by T lymphocytes. The pathogen's antigen are expressed on the cell surface or on an antigen-presenting cell. Helper T cells release cytokines that help activated T cells bind to the infected cell's MHC antigen complex and differentiate the T cell into a cytotoxic T cell. The infected cell then undergoes lysis. Adaptive immunity is of two types, humoral immunity and cell-mediated immunity. Adaptive immunity classified into two, humoral immunity and cell-mediated immunity. Humoral in the sense provided by B lymphocytes can recognize protein, polysaccharide, phospholipid and nucleic acid antigens, can act against soluble or free antigens, provide immunity to extracellular bacteria, viruses and toxins, cause type 1 and 2 and 3 hypersensitivity. Cell-mediated immunity provided by T lymphocytes can recognize only protein antigens, recognizes antigen presented by APCs with class 1 or class 2 MHC molecule, provides immunity to intracellular bacteria, virus, fungi and protozoa, causes type 4 hypersensitivity, causes acute graft rejection. Then hematopoietic stem cell, multipotent progenitor, common myeloid progenitor, common lymphoid progenitor. In case of common lymphoid progenitor, you could see B cell, T cell or NK cell. In case of common myeloid progenitor, you could see megakaryocyte, erythrocyte progenitor and granulocyte macrophage progenitor. So megakaryocyte or erythrocyte progenitor is of two types, erythroblast and megakaryocyte. Erythroblast RBC, megakaryocyte, platelets or thrombocytes, cytoplasmic fragments derived from megakaryocytes. Granulocyte, macrophage progenitor, neutrophil, eosinophil, mast cell and monocyte. Next is about B cells. B lymphocytes have derived their name from the set of maturation from Bursa fabricis in birds. B lymphocytes function in the humoral immunity component of adaptive immune system secreting antibody. B cells are generally classified into plasmid, all which release antibody, and other one is memory cell. B cell development in the bone marrow. B regulates construction of an antigen receptor, ensure each cell has only one specificity, checks and disposes of self-reactive B cells, exports useful cells to the periphery, provides a site for antibody production. Bone marrow provide a maturation and a differentiation microenvironment for B cell development. B cell maturation. The antigen-independent maturation of B cell occurring in bone marrow involve acquisition of B receptors, BCRs, co-stimulatory molecules, signaling molecules, co-repressors. Then the progenitor cells divide to produce pro-B cells, and pro-B cells differentiation is achieved by the interaction of progenitor with bone marrow stromal cells. The receptors involved in this interaction are adjacent molecules expressed by both the cell types, stem cell factor expressed on stromal cell and its ligand c-kit expressed by pro-B cell. The stromal cell secretes cytokine IL-7 which helps in converting pro-B cell into pre-B cell. The immunoglobulin gene rearrangement takes place at the pro-B cell stage. A successful rearrangement results in the expression of B cell receptor BCR on the surface of B cells. Then B cell receptor. The B cell receptor or BCR is a transmembrane receptor protein located in the outer surface of B cells. The receptor's binding moiety is composed of a membrane bound antibody that, like all antibodies, has a unique and randomly determined antigen binding site. B cell is activated by its first encounter with an antigen that binds to its receptor, its cognate antigen. The cell proliferates and differentiates into generate a population of antibody secreting plasma B cells and memory B cells. Function is to mediate internalization for subsequent proceeding of the antigen and presentation of peptides to helper T cells. BCR functions are required for normal antibody production, and defects in BCR signal transduction may lead to immunodeficiency. Then B cell epitopes. Function is to mediate internalization for subsequent processing of the antigen and presentation of peptides to helper T cells. BCR functions are required for normal antibody production, and defects in BCR signal transduction may lead to immunodeficiency. Then B cell epitopes, usually dependent on the native tertiary conformation of the antigen, protein folding, must be accessible, tend to be on the surface of the antigen, hydrophilic, may be made of sequential or non-sequential amino acid sequence. Epitopes made up of non-sequential amino acid sequences called a conformational epitopes. Binds to soluble antigen, no MHC molecule required. Large antigen contain multiple overlapping B cell epitopes. Then B lymphocytes have SIG molecules on their surface that recognize epitopes directly on antigens. Different B lymphocytes are programmed to produce different molecules of SIG, each is specific for a unique epitope. B cell activation in detail. B cells are activated when antigen binds to receptors on the B cell surface, followed by a co-stimulatory signal usually provided by a helper T cell. Antigens that require co-stimulation by a T cell to activate a B cell are T-dependent antigens and are usually proteins. In order for the helper T cell to stimulate B cell, both must be activated. This usually requires that the B cell internalize the antigen, process it, and then present it on the cell surface bound to the class 2-ML HLA molecule. The HLA antigen complex is recognized by a receptor on the surface of the T cell receptor or TCR. Then B cell activation, extracellular antigen, AGA fragment, HLA class 2 with the AG fragment, immunoglobulin receptors coating a B cell surface. Immunoglobulin receptors on B cell surface recognize and attach to the antigen, which is then internalized. Then HLA class 2 antigen fragment complex is displayed on B cell surface. Receptors on the T helper cell recognize complex of HLA class 2 and antigen fragment and is activated, producing a cytokines which activate the B cell. B cell is activated by cytokines and begins clonal expansion. Some of the progeny become antibody producing plasma cells. B cell differentiation. B cell development and differentiation occurs in multiple phases. The initial antigen-independent phase generates mature immunocompetent B cells that can bind to a unique antigen. The stage of development occurs in bone marrow and involves progenitor B cell proliferation and VDJ gene rearrangement, which produces clonally unique immunoglobulin variable regions that specifically bind the antigen. B cells complete further antigen-independent maturation into immunocompetent naive mature follicular B cells in the bone marrow and spleen. The antigen-dependent phase of B cell development occurs following B cell activation by antigen binding and co-stimulation. These signals promote B cell differentiation into either memory B cell or terminal antibody-secreting plasma cell. The antigen-dependent phase of B cell development involves activated B cell proliferation, antibody affinity maturation, and antibody class switching, all of which occurs in the germinal centers of secondary lymphoid tissues. Multiple cell surface associated proteins and transcription factors are required for each stage of B cell development and differentiation. Then about T cells. T cells are part of the immune system that focuses on specific foreign particles rather than generally attack any antigens. B cells circulate until they encounter their specific antigen. As such, T cells play a critical role in immunity to foreign substances. Types of T cells: T helper cells, cytotoxic T cells, regulatory T cells, formerly known as suppressor T cells, memory T cells, helper T cells, CD4 positive. Helper T cells release molecules, cytokines, into the bloodstream to warn the immune system of the presence of a dangerous cell or virus. Cytotoxic T cell, CD8 positive. Cytotoxic T cell attack themselves to other cells in the body. If the cytotoxic T cell learns that the cell is diseased or pathogens, it will be one of two things: force the cell to commit suicide, or apoptosis or secrete enzymes that will kill one cell. T cell receptor. The T cell receptor or TCR is a molecule bound on the surface of T lymphocyte or T cell that is responsible for recognizing antigen bound to major histocompatibility complex molecule. Structure of T cell receptor. Heterodimer, alpha plus beta chain linked together by a disulfide bridge. Constant region anchor the receptor to the plasma membrane. The T cell receptor or TCR is a molecule bound on the surface of T lymphocytes that is responsible for recognizing antigens bound to major histocompatibility complex molecule. Structure of T cell receptor. Heterodimer, alpha plus beta chain linked together by a disulfide bridge. Constant region anchor the receptor to the plasma membrane. Then constant region will be there, anchor the receptor to the plasma membrane. Variable region is also there, contact the antigen. Each T cell bears TCRs of only one specificity, allelic exclusion. Both alpha and beta chains have a variable V and constant C region. V region of the alpha and beta chains contain hypervariable regions that determine the specificity for antigen. Alpha chain, beta chain is there, variable region V, constant region C, hinge H, transmembrane region, cytoplasmic T. T cell epitopes. Involves tertiary complex. T cell receptor, antigen and MHC molecule must be accessible, tend to be on the surface of the antigen, hydrophilic. May be made of sequential or non-sequential amino acid sequence. Epitopes made up of non-sequential amino acid sequences are called conformational epitopes. Bind to soluble antigen. No MHC molecule required. Large antigen containing multiple overlapping B cell epitopes. Then T activation and mechanism. T cells originate in the bone marrow but are matured in the thymus. However, they are not activated until they find their specific antigen. They bind to this antigen on the surface of antigen-presenting cells. Typically, several types of T cells are involved in this, mainly C4 helper T cells and CD8 cytotoxic T cells, and together they form the MHC complex. The activation of T cell is not always as binding to the MHC. Both helper T cells and cytotoxic T cells, two of the types of T cells, need secondary signals to become fully activated and be effective towards the threat. These are provided by several molecules such as CD28, which activate helper T cells. In general, there are three types of T cells: cytotoxic, helper, and regulatory. All of these must react to foreign antigen strongly to be effective for immunity. T cells with a strong reaction are also given survival signals by several molecules such as ICOS and OX40. These are only expressed on the T cell surface following binding of the antigen to ensure it is only active after it responds to a pathogen. After activation, communication occurs in the form of cytokines. The cytokines decide what form of responder the cells turn into. Helper T cell become Th1, Th2 or IL-17 types. Each of these types has its own role in the continued development of further immune response. T cells mobilization when they encounter a cell such as a dendritic cell or T cell that has digested an antigen and is displayed antigen fragments bound to its MHC molecules. Cytokines help the T cell mature. The MHC antigen complex activates the T cell receptor and the T cell secretes cytokines. Then NKT cells, natural killer cells. Natural killer T cells are a heterogeneous group of T cells that share properties of both T cell and natural killer cell. Most NKT cells express a semi-invariant cell receptor and some express CD4. Many of these cells recognize the non-polymorphic CD1D molecule, an antigen-presenting molecule that binds self and foreign lipids and glycolipids. Then NK cells, natural killer cells, are a type of lymphoid cell, innate lymphoid cell, which function in the innate immune system to remove infected or cancerous self cells derived from common lymphoid progenitor but express antigen-specific receptors. They are subdivided into ILC1, ILC2, ILC3 tissue resident cells, distinguished by the secretions of their cytokines. Developed in thymus and distinct from cytotoxic T cells, play a significant role in destroying cells infected with intracellular pathogens. NK cells destroy the late target cell not by phagocytosis but by releasing biologically potent molecules. NK cells are perforin and granzymes. NK cells use perforin and granzymes to kill their targets. NK cells differ from TC cells in that they kill targets in antigen non-specific manner. Shediac-Higashi syndrome, an Archibald pronunciation, an autosomal recessive disorder is associated with the lack of NK cells. CHE-DIAK Higashi syndrome. Thank you for listening.